You're watching EASD TV. Now, when I see chronobiology in a program, I'm there like a shot. It's such a fascinating area. And to tell us more about that session, we've got two people who were presenting at it. We've got Charna Dibner from Geneva University and Frank Scheer from Brigham and Women's Hospital, although you come via the Netherlands.
Correct.
So just outline for us what we mean by chronobiology.
So chronobiology really deals with the importance of rhythmicity in our physiology and our behavior. And specifically here, we're talking about circadian rhythms, meaning circa a day, so approximately a day. And we basically have an intrinsic clock in our body, and in fact, these are multiple clocks in our body in virtually every organ and tissue.
And indeed every cell.
And in every cell. So we have the same underlying molecular machinery that influences the function of those organs and tissues. And so because of that highly preserved mechanism throughout evolution, this is important in driving a lot of our bodily functions throughout the day and the night, and plays a very important role, which we can understand especially when these rhythms become perturbed.
Now, our circadian rhythm is set by light.
It's synchronized by light, correct?
Synchronized by light. So are we, just as we go around our daily lives, are we sometimes out of sync with our clocks?
Unfortunately, yes. Certainly in modern day society, there are so many factors in our lives that are very frequently misaligned. So we can think about artificial light in the evening hours. We can think about eating snacks late at night when we really should be fasting. We can think about the jet lag that many of us are experiencing here at this international meeting.
Sorry about that.
And so indeed, modern lifestyles, we believe, are playing a role in the increasing prevalence of type 2 diabetes as well.
Now link type 2 and the clock for me.
Right, so there are a lot of links in there. One of the most fundamental is that our metabolism is very tightly intertwined with the circadian system, meaning that the circadian system influences our metabolism, and our metabolism feeds back on our circadian system. And examples include, as is relevant here today, is the importance of the circadian system in glucose control, both on the pancreatic beta cell function, as well as also insulin sensitivity. And so those factors are influenced every day in our lives by the circadian system.
So what was the subject of your presentation?
So I focused on a few things, two primarily. One is whether the well-known day-night rhythm in glucose tolerance is fully explained by our sleep-wake and rest activity cycles, or that there might be a role for this internal circadian system. And so we provided evidence that in complicated experimental human trials under highly controlled laboratory conditions, we can tease those components apart and show that indeed the internal body clock plays an important role. Then I spoke about one of the factors that we believe and have shown is involved in this communication from the circadian system to glucose control, which is the hormone melatonin, which typically is high at night. And what we have shown is that there are large inter-individual variations between the tolerance to late-night eating that can be pinned to genetic variants of this melatonin receptor. And so this is a very common receptor variant referred to. The gene is MTNR1B. And then in the later stage of my talk, I talk about the well-known observations that in chronic shift workers there's increased risk for diabetes, and not just diabetes, cardiovascular disease, obesity, cancer, etc. And so what we addressed is the question whether This might be explained by differences in work stress or dietary habits or socioeconomic status, or that it might actually be something about the fact that our sleep-wake and eating-fasting rhythm are misaligned, so out of whack with this internal body clock. And so we've provided evidence, again using highly controlled experimental studies within the same individuals, align and misalign, and also showed that this is relevant in chronic shift workers. And then finally presented on data, which is the next step now, is what can we do about it? Can we develop countermeasures? Can we mitigate these adverse effects? And how do we do this? And we're kind of, in that way, combining those two insights using meal timing and circadian misalignment to see if meal timing, if we correct the timing of food relative to our circadian system, if we can prevent the adverse effects. And so we've some exciting data, I think, proof of principle data showing that correcting circadian meal timing can greatly reduce the adverse effects on glucose control and many other aspects.
So I want to come back in a moment to some specific recommendations that people might find helpful to pass on to their patients. But let me go to you, Charna, because you've been looking at the islet cells in vitro and looking at their clocks. And how does this marry up with the work that's just been described to us?
Yeah, thank you, Vivian. So as Frank just mentioned, these clocks are cell autonomous, right? So this fundamental discovery was done in Geneva in the lab of Uli Schiebler over 20 years ago, and I was privileged enough to be trained there as a postdoc. So basically you have this perpetuum mobile, this molecular mechanism in every, as you said correctly, single cell in the body. And that allows us, so what was different in our work, that allows us to really zoom into molecular clocks in human pancreatic islets. And that was the first part of my talk. And this is very complementary to what was Frank Scheer is doing, for example. So also, if you think about human pancreatic islets and circadian studies, to get the circadian rhythm, you have to do repetitive sampling across 24 hours by definition, because circadian come from Latin circadian, right, about the day. So contrary to normal scientists, we have to do everything across 24 hours at least, and Frank is doing it across the week or 2 weeks, right? So for that, you cannot do it repetitively in human pancreas, right? So that's why this in vitro, as you said correctly, was really fundamental, and that is based on Schiebler's discovery. So what we set up was first observing this molecular clock in human islet cells and separately alpha and beta cells. We did this specific labeling with fluorescence, and we could really follow these rhythms with very high time resolution. So now why EASD would be interested in that? Because along with the molecular clocks, we also followed 24 hours secretion of insulin and glucagon, and that was basically the first link and first demonstration that these hormones are secreted cell autonomously by human islets in a temporally regulated circadian manner. What also we did was, so we could manipulate the clock. Basically, we did circadian misalignment in the dish. So we introduced inhibitory molecule for one of the clock components called clock, and we basically disrupted the clocks. And that was concomitant with disrupted insulin secretion. You have less secretion of insulin by the human islet if the clocks are perturbed. And that brings us to type 2D situation, where we actually discovered, so that was our recent publication, that does not come as a surprise that, of course, human islets from type 2 diabetic donors secrete less insulin, but what we showed that they also secreted on a perturbed 24 hours manner. And what was reasonably interesting, I think, that when you restore these clocks, at least in vitro, you could also restore to some extent basal and induced insulin secretion. So that was
that's fascinating. And there was also a third presentation, I think. I know that the presenter is unfortunately not able to be here. Can you just briefly take us through that, very briefly?
Yeah. So Christian Benedict gave the third presentation, speaking about the importance of sleep on glucose control. And so one example he gave was the importance of a very common sleep disorder, which is obstructive sleep apnea, which is when your upper airway closes at night when you sleep repeatedly overnight. And it's very disruptive to the sleep. It causes hypoxia and sympathetic nervous system surges. And experimentally, for example, if you induce this in a person, you can see very quickly changes in the uptake of glucose during a euglycemic hyperinsulinic clamp. So really showing experimentally that very prevalent sleep disorders, and by the way, obstructive sleep apnea most commonly is undiagnosed. So most of the people who have it don't even know about it.
Not by wives.
Right. So the wives may notice that the husband snores. If the couple is lucky, one of them recognizes that this is an increased risk factor for obstructive sleep apnea.
I should say it's not always in men, by the way. Just in case anybody's going to write in.
Right. And so, for example, this is how I recognized that my young son, when he was about 4 or 5 or so, had obstructive sleep apnea, because I heard his breathing stop for more than 10 seconds, sometimes 30 seconds. And so just to point out that and this is just one example, but sleep is really important for glucose control as well.
Yeah, and we see it disrupted in, for instance, things like Alzheimer's. That's where the sleep cycles, whether it precedes it or it's caused by it is still, I guess, a debate. Yeah. But it's
there's good evidence that there are changes in the circadian system that happen even before the onset of Alzheimer's disease and dementia. So the other thing, now that you bring up Alzheimer's disease, is that sleep is also very important in clearing toxins from the brain through the glymphatic system that's regulated, which is basically the brain equivalent of the lymphatic system. So it's a clearing mechanism that is stimulated during sleep. So just to say that, yes, to follow up on what you're saying, sleep and circadian rhythms both are very important for many aspects of our health.
Okay, so let's go to the practical implications now. You were talking about trying to resynchronize the external cycle with the kind of behavioral cycle. Tell us a bit more about that.
Yeah, so I think one important observation we made was that when melatonin levels are elevated and concurrently you would eat, that your glucose tolerance is relatively impaired.
So your melatonin levels would be great because it starts to secrete about 10 o'clock at night at your normal bedtime.
So on average it starts to increase about 2 hours before your habitual bedtime. There's about a plus and minus 2 hour range looking at inter-individual variability. And so that is one mechanism. The other is that many individuals are orally taking melatonin. Sometimes for sleep, sometimes because of jet lag, etc. And so also there, if you take it exogenously, close to a meal, we've also shown that this impairs glucose tolerance. This is work together with Marta Garoulet. And then we have found that not everybody is impacted in the same way. And so if you are a carrier of the risk variant for diabetes that is residing in the MTNL1B gene, which is one of the high affinity melatonin receptor genes, then you are even more at risk for having worsening of your glucose control. So the simple conclusion one could draw from that would be, don't eat close to your elevated melatonin, either close to your bedtime or after oral intake of melatonin.
So all those things that your old wives' tales about eating late at night and don't eat cheese at night, I mean, I'm sure there's a different one for every different version of that.
Swiss. Unless the cheese is Swiss. Right?
Unless the cheese is Swiss, of course. There's a variant in every culture that says don't eat late at night, and actually it turns out to be true. It's really important.
So right, and so I think that's an important message here. The other would be that there's also hope, in the sense that if you now redirect the timing of your meals, you can actually leverage this system to improve health overall. And so the example I gave was in simulated night work conditions, which a large portion of the population, about 20% or so, is engaged in. And so these people experience this chronically. Many of them have no alternatives, and so it's also on the rise still. And so this is really a critical problem in the field, where now these type of studies provide some early hope.
Fantastic. Well, thank you so much for coming to talk to us about this. It's such an interesting area, and I suspect there's a lot more research to come here.
Thank you very much.
Thank you so much. And that's all for the moment, but we'll be back soon.